Right, so this video is unit 3.2 on circulatory systems, comparing single circulation, double circulation, open, closed. We're going to be looking at the differences in the artery, vein and capillaries, and we're also going to look at the movement of blood, and tissue fluid, and seeing how that works between the arterial and venous end. Okay, another video I'll be looking at the heart structure, but that's the second half of 3.2, and I'm going to split that into two videos because I do like to ramble on a bit. Okay, so first of all we're going to look at what makes an effective transport system. We need a fluid medium to carry nutrients, so that's going to be the blood. So what else do we need? We need a pump, and this pump is going to generate a high pressure. So that a pump creates pressure and this moves the fluid around, so it moves the blood around. And for us we've got a heart, and for insects sometimes they use their muscular pump, or they even use these specialised organs that use peristalsis to move the blood along, and we'll look at that later. One of the things effective transport systems need is a good exchange system, so good exchange surfaces. So we've got capillaries, so if we was going to look at the lungs, we've got the capillaries and the alveoli. They're good exchange surfaces, but I won't go into too much depth here, that's a different unit. And we need tubes or vessels to move blood around by mass flow. Okay, so vessels are important, otherwise we just have blood sort of in an open circulatory, but even worse, the cells just bathe in the blood and it'd be really inefficient. Right, so we're going to look at the two types of circulation. I've got single circulation and double circulation. I've took these diagrams off Google, credit to whoever made them. I thought I'd spare you my drawings today, although you've got some coming later on. Single circulation we'll look at first. It is when the blood flows through the heart once for each circuit of the body. So as we can see, this example is a fish, and the blood just circulates through the gills and to the rest of the body here, and it just keeps circulating round and round. There's no two circuits, so if you think surely that's what we have, we've got lungs and we've got the rest of the body. Ours operates as a double circuit, so we have a pulmonary circuit where the blood's pumped to the lungs, it comes back to the heart, and then it's pumped back out to the body. In here it goes round, it goes to the lungs and body in one complete circuit. Okay, in single circulatory systems you get a low blood pressure in the gill capillary. So we're using a fish as our example here. We get a low blood pressure here, and what we know about that, low pressure means that we have decreased rate of diffusion. So it is harder for fish to, I didn't know I could do that with my arrows, for fish to move solutes out of their blood into their cells. Okay, I'll move that arrow around because I can now. And another thing is a low blood pressure. Single circulatory systems just have low blood pressure in general. So low blood pressure going to the body, the blood doesn't move that fast, and once again we have a low rate of diffusion even at the body, the systemic bit, so that usually means to the body. Overall we've got low gas exchange, we've got reduced oxygen delivery, reduced carbon dioxide delivery, and the removal of urea and metabolic waste is really low. So basically single circulation is not that efficient, and our more efficient one is double circulation, and that's what we've got. So I think we'd all like to consider ourselves as more efficient than fish, and this is probably the basis of it all. It's our double circulatory. So our example is humans. And double circulatory systems, as we've said before, the blood's pumped to the lungs, it comes back to the heart, and then it is pumped through a different circuit, the systemic circuit, to the body. It doesn't go completely around. So some advantages of this is it allows pressure to be varied. So you can have a low pressure at the lungs, which for us is helpful because the alveoli, remember, they've got delicate lung capillaries. These are really narrow. They're just one red blood cell wide, some of them, and they're really delicate. So a low pressure for us there is quite handy because our alveoli are so specialised, we don't really need them to be too efficient because we've got other means to create that efficiency. So we've got alveoli and we've got the moisture, so oxygen diffuses in, but that is when we look deeper into the lungs, which we're not right now. Another advantage is there's fast removal of carbon dioxide and urea and fast delivery of oxygen, so high gaseous exchange. So whereas in single circulation we have a low gas exchange, here in double circulation we've got a high gaseous exchange. Because we're allowed to, in the systemic circuit, we can have a high blood pressure, and this creates bigger differences, bigger gradients to cause concentration gradients, is a better word, to cause solutes to move in and out of the blood. Okay, and that's our last point. A high concentration gradient is maintained. If we had lots of waste coming back from the lungs to the body, we would not be able to load the waste on because the waste would not want to move themselves into the blood because it's already got a high concentration in the blood. We know that things move from a high concentration to a low concentration, and if we've got a high concentration of waste, the waste is not going to move out equally. If we've already got a high concentration of oxygen in the blood at the lungs and not got deoxygenated blood going to the lungs, we're not going to get oxygen moving in to our blood, and then we're just going to be in a constant cycle where we get less and less materials. In open circulation, blood is not always held in the vessel. So an example of this is our insect here. Blood circulates through the body cavity, tissues and cells, bathed directly in the blood, whereas in closed we have something called tissue fluid. So if I just draw an example of that for you on here, we've got a cell there, and in between them we've got blood. They're bathed within that blood, okay, rather than in the closed circulatory system. I'll draw this in a different colour. We've got a cell there and a cell there, and they're bathed in tissue fluid. So I'll do tissue fluid in yellow, okay. But they're really messy. I'm going to get them off the screen because they look ugly. Okay, so open circulatory systems, we've got cells bathed in blood. Some animals require muscular movement to pump their blood, like we said before. So this muscular movement can be generated by flying. Hence, some insects constantly have to be moving. Right. Some insects also have specialised organ that's not a heart. It's basically a tube, a muscular tube around the vessel that contracts to squeeze blood. So you just have blood in the vessel and it's got a muscle around it, and via peristalsis. You might remember that in our esophagus. We don't learn it, but we did it at GCSE, where that contracts by peristalsis to move the bolus of food down. Here we've got peristalsis to move blood along, so a ring contraction moves down and pushes blood along. In open circulatory systems, we've got a low blood pressure, so that causes blood flow to be really slow. We're going to now look at closed. So here we've got the main point: blood stays in the vessels. That's the point of closed. Tissue fluid instead of blood bathes the cells. By keeping the blood in the vessels, we can generate a high pressure and rapid oxygen and nutrient delivery also we get a rapid removal of waste products. It's not really too complex, these two topics. You just have to remember, insects is a good example, where the blood is not always contained in the vessels, and any sort of normal mammal, worms, any animal really, it's not like insects, they have closed circulatory systems. Right, now we're going to compare our arteries, veins, capillaries, arterioles and venules. Okay, and there's, once again, I've stolen this off the internet, credit to Pearson Education 2011 who've copyrighted this image. Hopefully they won't sue me for this. Right, first of all, we've got our 3 layers. We've got the tunica intima, which is a thin layer of elastic tissue. It allows the walls to stretch and recoil to help maintain blood pressure. We've got our tunica media, which is a layer of smooth muscle to withstand high pressure. Okay, and we've finally got our tunica externa, which is a thick layer of collagen and elastic fibres to provide strength to withstand pressure and recoil to maintain pressure. So it's tunica intima, tunica media, tunica externa. No excuses, you've got to learn those phrases. I've been caught up in a paper before for not learning them. So now let's compare the artery to the vein. So artery, first of all, they've got a really small lumen. As you can see here, I might actually draw that onto the diagram here with an arrow. They've got a really narrow lumen. Small arrows, you can see I've just drawn there. This creates a really high pressure in the artery. As remember, arteries taking blood away from the heart, it needs to be at high pressure because they've got a long distance to transport this blood. Okay, they've got thick, smooth muscle, so a thick tunica media. Okay, and let's remember, our tunica media is the smooth muscle layer. That's to withstand pressure. Like we've said, they're dealing with a high pressure, so they've got a thick tunica media shown here on the diagram. It's really thick to withstand that high pressure. They've got a thin tunica intima. Okay, so that is our thin layer of elastic tissue, allowing stretch and recoil. So arteries don't really need to recoil that much because they've generated the pressure here. Veins, however, when we look at it in a second, they do, so they're going to have a thick tunica intima. But lastly, for arteries, they have a thick layer of collagen and elastic tissue. That is for strength and support. So our tunica externa here, just shown here, is really thick, contains lots of collagen because we're dealing with high pressure. I think I should write that down. High pressure in our arteries. Okay, we're dealing with lots of pressure, so we need lots of collagen, thick tunica externa, and we need lots of thick smooth muscle in our tunica media here. But our tunica intima in the arterioles are small. So let's draw some arrows next to it. For artery, we've got a thick, so I'll draw a high arrow. Tunica externa is thick, but our tunica intima is not so. Okay, we've got that narrow diameter. In a vein, let's have a look at our lumen. It's much wider. Even though my arrows don't really show the difference, as we can see on the diagram, which is why I didn't draw it, it's much bigger, it's much larger. So we're dealing with a much lower pressure, okay, in our vein. This is the large lumen, they've got a thin, smooth muscle, so a thin tunica media layer. Okay, they don't have to withstand any high pressure. So once again, that means they'll have a thin tunica externa. If you've got a thick tunica media, you're likely to have a thick tunica externa. They sort of do the same sort of jobs, provide strength and support, just help withstand pressure. Okay, so let's draw our arrows here. Already we can see a difference. Our arteries are the opposite. They've got a thin tunica externa and a thin tunica media because they've got a low pressure, which I'll also write here. Low pressure. Right, our last one to look at is our tunica intima, which you guessed it is once again going to be the opposite. We have a thick tunica intima. This is because arteries need to elastic recoil for venous return. So venous return is when you're returning blood back to the heart. It's a topic not covered in too much depth in biology. I do PE, and we used to study that a bit more at AS. Just dropped all my coins on the floor. We need to be able to get the blood back to the heart without pressure. So elastic recoil, like contraction of the veins, helps to generate pressure and push blood up. Then we've got valves here, which were not, that was a bag to highlight, which were not present in the arteries. We can now look at capillaries. Okay, so capillaries have very thin walls. Okay, because obviously this is where diffusion takes place. We're transporting substances here between like the cells and capillaries, exchanging nutrients, exchanging waste. It takes place here. So capillaries, they've got very thin walls. So a single layer of flattened endothelial cells, which are these cells here, which is the inner layer. Also here, it's found in all vessels. Okay, this endothelium layer is the inner layer, and capillaries just basically have endothelium layer and then their basement membrane. They've got leaky walls, okay, which allows blood plasma and dissolved solutes to leave the blood, and they've got a narrow lumen which can be as small as one red blood cell, such as in the lung capillaries, so that's 7 micrometers, 7 um. Red blood cells are squeezed against the capillary wall, which helps to transfer the oxygen and reduces the diffusion distance, as well as increasing the resistance and reducing the rate of blood flow. So the narrow lumen is really important here. Reduces the blood flow, so more diffusion can take place. It increases resistance and overall decreases the diffusion pathway for dissolved minerals to go through. So does the thin walls. Okay, I've pretty much overkilled that, but we're going to look at arterioles and venules. So arterioles, it goes from arteries to arterioles to capillaries. So they're the in-between layer. They've got a layer of smooth muscle that can constrict and reduce blood flow, and they're used to divert blood flow to areas requiring oxygen. So they're slightly smaller than arteries. They're the next stage. So we have a big, large artery, then the blood goes into arterioles, and then to capillaries with an even lower pressure, as we can see in this diagram here. These being capillary network in the middle. Then we've got venules, which are just before veins. They're a thin layer of, they have a thin layer of muscle and elastic tissue and some collagen, and the blood flows through the venules into the vein. So blood flows through the arteries, then the arterioles, the capillary, the venules, then the vein. Okay. one thing here is this diagram here, which we're now going to look at and see how solutes are transferred here. So I've got some of my notes here in the top left hand corner, and it's just a little table to show what's in blood, plasma, tissue fluid and lymph. So first of all, hydrostatic pressure in the blood plasma is high, in the tissue fluid it's low, and lymph is low. That will be important in the next one, and I'll just ignore oncotic pressure for now. But what we want to look at here is the cells. So we've got red blood cells, neutrophils and lymphocytes in the blood. Only have neutrophils in the tissue fluid, only have lymphocytes in the lymph. So lymphocytes, easy to remember. And fats wise, transported in lipoproteins. In our tissue fluid we've got a few fats, but in our lymph we've got a lot of fat near the digestive system. Okay, that will help you understand what's going on here a bit more here. So first of all, we've got oxygenated blood coming from arteries. It enters the capillary network through arterioles into our capillary network. And here at the arteries we've got a high hydrostatic pressure. So blood plasma is forced out. Okay, let's do this in nice orangey colour. Blood plasma, oh, that's probably too thick. We'll use this. Blood plasma basically leaves this capillary network and bathes these purple cells that I've drawn. Okay, so that looks like a squiggly mess, but that is representing blood plasma leaving and bathing this capillary network. So the plasma leaks into capillaries, it surrounds the cells and tissues, it carries dissolved nutrients and waste with it, and it moves by mass flow. Okay, so if plasma is forced out at the capillaries, the blood is under high hydrostatic pressure. So then we've got the capillaries. The dissolved nutrients move out with the plasma. However, the red blood cells, platelets and white blood cells remain in the blood. As we said here, we don't have any of those cells in the tissue fluid, just neutrophils. And then nutrients are exchanged with the cells. So our cells in purple here, they take oxygen from the plasma and they put waste back into the plasma. Now let's get rid of all the plasma we've drawn there. As we go down to the venule end, we've got a low pressure because we've got our wider veins here, a much lower pressure here. When we're talking about pressure, we're talking hydrostatic pressure. So plasma, which was accumulated here, then draws back in to the blood capillaries. Okay, so tissue fluid, which is basically blood plasma, carrying loads of waste now. Okay, deoxygenated blood plasma moves back into the capillary network, and then it moves through the veins as deoxygenated blood, and they go back to the heart, so it can become oxygenated again, and that repeats. Excess tissue fluid is drained by the lymph, and the lymphatic system then has lymph vessels. They return the blood, they return all this excess nutrients and stuff back at the lymph vessels which connect with blood, and the lymph contains more lymphocytes. The lymphatic system is involved in unit 4.1 when we're dealing with infections. Right, finally we've got this diagram here which I've drawn nicely, and this is to represent why solutes move out of the blood at the arterial end and back into the blood at the venous end. And there's two key words here. First of all, we've got hydrostatic pressure, which is the force exerted against the sides of the vessel. So hydrostatic pressure, as I'm going to do in green, is always pushing outside against the vessel. So if I draw an arrow, it's always going that way. Oncotic pressure is pressure created by the osmotic effect of solutes. And as we've written here, such as, just to put this in perspective for you, plasma proteins draw in water as they cause the blood to have a low water potential. So hydrostatic pressure is always pushing outwards. Oncotic pressure is the drawing in of water by these plasma proteins. They want to draw in tissue fluid back into the blood. Okay, let's get rid of those two arrows just for now. So let's look at our arterial end, and here we've got a high hydrostatic pressure. So if we've got a high hydrostatic pressure, we've got a lot of force wanting to go this way. Okay, so as we see here, 4.3 in the arterioles. This is in kilopascals, kPa. I'll just draw kPa here so you remember. That's the units we're dealing with here. Hydrostatic pressure is the force exerted against the wall. It's going outwards. Basically, our plasma wants to leave because there's a high hydrostatic pressure. The oncotic pressure here is from minus 1.3 to minus 3.3, and that is consistent. It's always there. Minus 1.3 kilopascals to minus 3.3. That's the drawing potential of these plasma proteins, and that's a smaller arrow. So there's a smaller difference there. So basically, our net outflow is 1.2 kPa, kilopascals. And that is the difference between, I'll put net outflow, 1.2 kPa. And that is the difference in these two pressures. So we've got 1.2 kilopascals more going outside of the capillaries than we do going inside. So blood plasma drains out at the arterial end. Now let's look at the venous end. So here we see 1.6 to 1.1. We've got a much lower hydrostatic pressure. So let's do our hydrostatic pressure arrow. We know it goes out this way. It's much lower. Okay. And our oncotic pressure remains the same. Remember, our plasma proteins are always in the blood. They always draw the same amount of energy. However, in comparison now, we've got a net inflow of blood. So blood, oops, the wrong one. Blood plasma. So we've got blood plasma carrying all these waste materials at the venous end, and our net inflow is minus 1.5 kPa, kilopascals. So the plasma returns back into the blood at the venous end. Minus 1.5, why is that? It's the difference in these two. So we've got 0.5, one difference going down. We've got a 2 difference going up. So 2 minus 0.5 is minus 1.5 to go back into the capillary. So in summary here, we've got a higher hydrostatic pressure than oncotic pressure, hence the net outflow is out of the blood, but at the venous end we've got a lower hydrostatic pressure than we've got an oncotic pressure, hence the net inflow is into the blood. So blood moves along from the arterial end, we get rid of our oxygen and nutrients, and we gain waste material here at the venous end as it goes back into the blood. Okay, that is a summary on circulatory systems. Next video I'm going to look at the heart structure. I'm going to also look at the different pressure differences within the heart and ECGs, blood transport. So we're looking at the transport of oxygen and carbon dioxide. That is the second half of unit 3.2. Okay, that'll be next video. Thank you for watching.